What I will be talking about today is our bodies, but especially all of the species we interact with, that our bodies interact with in every room that we're ever in. And I know everybody always hopes for resolutions in talks, but that's not the kind of talk I ever give. I always leave a talk with more mysteries than resolutions. It's the kind of person I am. And the books outside are 3 of these books. This 4th book comes out in March, but what I'm talking about today is actually building on the wildlife of our bodies and then moving into what's a new book. Let's see if I can get this to go. That's a lovely tapeworm that's named after a chancellor at the University of Connecticut. The tapeworm biologist and the chancellor differ as to whether that was flattering. Oh, shoot. I'm just going to click it. The reason I want to think about tonight what we live with inside is that it turns out that that's a hugely important part of what we're exposed to. This chart shows the geographic area, the proportion of Earth's terrestrial area composed of different biomes. What you see at second to the top is that the indoor biome inside buildings is now larger than tropical and subtropical coniferous forests as a biome. And it's rapidly growing. And this is my paper, and so I know where the data come from, and I can say that that's mostly based on the first floor. It's not, we don't even include that buildings go up, which is to say that that realm is huge and growing, and it's where we spend virtually all of our time. Think about how many hours you spent outside today. For most of you, it's probably very, very few. And we can look at this in the context of particular cities, too. And so this is Manhattan. Let's see if this works. And there we go. And so you can see in Manhattan the population in millions, but then also how the indoor biome grows. The top right is now the area of the indoor biome in Manhattan, including different floor levels. The circle on the left is the land area of dirt in Manhattan. And so in Manhattan, the indoor biome now vastly exceeds the outdoor. And so that's a weird moment in history. And so for me, that really begs the question of, as we think about that indoor biome, which species should we live with? Which species benefit us? And are we making choices that favor those species? As this cat, I think, offers up, we don't do a great job of choosing the species we live with, typically. This is just what happens when you Google cat picture. That was the first one. But my point tonight is to consider what we should live with and what we do live with. And it turns out we don't actually know as much about what we live with as you might think. This is a field that started a long, long time ago, and so the first studies ever of what lived in houses were from the late 1600s by an amateur scientist named Leeuwenhoek. This is the microscope he used. These are some of his amazing drawings. But then what happens is germ theory comes along. And so whereas Leeuwenhoek thought these things were marvelous and beautiful and he studied them all, germ theory changed how we thought about all of this. All of this became bad. And so it became part of the long narrative in which humans kill stuff. And so the idea that there might also be good stuff really was left by the wayside. Until very recently, when we started to be able to look. Oh, that's too gross a Q-tip, isn't it? I didn't think it'd be quite so big. And so what happened recently that changed everything is that we developed new techniques for studying what we live with. And so we can study both things that we know how to grow, how to study, how to keep in the lab, and things that we can only know are present based on their DNA. And so we can use new technologies to take a swab like this of your ear pit or of a little bit of dust, and then we can look at all of the DNA present on that swab. I can tell you in advance as a kind of spoiler that a swab like that, if it were the dust off the back of your seat, will have no fewer than 1,000 species in it. And so what are those species and what do we know about them? Well, first of all, I thought that was my phone, but it actually wasn't. I did okay. We know that the habitat in that dust is actually really complicated. And so we might imagine there's a lot going on. And we know that the habitat of the whole home is kind of complicated. This is a heat map of a house, which makes clear that different rooms are different. And so what we did is that we engaged the public to study their own homes. And so we gave people swabs, and we said, swab these key habitats. These are all people who swabbed their homes, 1,500 homes in total. And from each one of those swabs, see, that dust mite should look cool that big. Yeah, no, that does. We looked at the bacteria, the archaea, which are another kind of single-celled life form that we usually think live in deep-sea vents, the fungi, the plants, and now we've actually started to look at the animals, all based on those swabs. This makes our lab look like Santa's workshop a lot of the time, which not everybody is equally appreciative of. The elves are the ones who don't like it. And what I'm going to talk to you about today, to narrow down the scope a little bit, is the first part of this, not the full 1,500 houses, but the houses we started in Raleigh and Durham, which include a bunch of deans' houses, and so it's fun to know what lives in a dean's house. I've got jokes there, but I'll let them be. And so we had people swab these habitats. And I trained working in tropical rainforests, and so I was thinking, like, what are the habitats in a house that are kind of like different habitats in a rainforest? And so these were the ones we thought about. And so people swabbed like this. And what we found were thousands of species of bacteria, even in those first houses. And that's not to mention the rest of life. And we found it was incredibly strongly structured. And so you might not have seen one of these before, but this is a kind of map. And the points are closer together. Each point is a sample. If they're more similar in terms of what was on that sample, they're closer together. And so the blue dots are all close together, and they're all sort of outside samples because there are a bunch of soil bacteria on them. Does that make sense, you guys? Yeah. And then everybody's okay with that. And then the stuff down here, this is sort of mostly food-associated microbes. The purple dot is somebody's TV that had like a hot dog on it. Microbes tell things we don't always want to know. And the top right is where we can't totally tell toilet seats and pillowcases apart. It's just biology. And so we can do this kind of work indoors, much as we would have done it in a rainforest. We can then go house to house and look what explains differences from one house to another. It turns out that the biggest single factor that explains what lives in your house is what kind of pets you have, and that dogs have a huge impact. That there's a bunch of stuff, and I won't go into this in detail, that's way more common on TV screens and pillowcases in houses with dogs. And that it's on the TV screens means it's getting everywhere up into the air and floating around. And some of that's soil microbes, some of that's saliva, some of that's dog feces. Less dog feces than kid feces, so it's okay. And what we found in total is that we can predict a lot of things about which microbes you have based on what you do in your home. So the ratio of women to men is a really big effect. The types of pets you have, cats matter too. The food you eat, so we can actually pick up if you ate a crab in your kitchen. We did this in Colorado, and this is before marijuana was legalized everywhere, and we got weed and DNA floating around the houses. How ventilated your house is and how it's ventilated. But there's something else here which really sort of tipped the scales in how we thought about this, which is that as we looked at other kinds of life, we realized we were only seeing a teeny piece of the picture. And so this is the number of kinds of species, basically, of archaeans we found in houses. This is the number known from Earth when I was a graduate student. And I know for the kids in the front, I look really old, but I'm not that old. This is the number we found in people's houses. When I was a graduate student, they mostly lived in deep-sea vents or like the hind end of a cow. We now know they're in freezers, too. Bacteria, we're now up to almost 100,000 species from inside houses. That's a lot. But the one that really floored me is fungi. And so if you look at these three lines, the middle line is the number of species of fungi we found indoors. Oh, you can't see the X here, sorry. And it's just as a function of the number of houses we've sampled. And we've found more kinds of fungi inside houses than there are named kinds of fungi in North America. And so earlier, some of the 10th grade students were asking me, well, what can you still discover? There are more kinds of fungi in your house than we've yet named, which is to say finding new species is as easy as sitting on the toilet or opening your fridge. And then animals, we've now found thousands of kinds of animals living in houses. Many of them we know very little about. And so all of this begs the question, what's good and what's bad and what do we want in our houses? And what's amazing to me is that this seems like a really obvious question, but we've basically never asked it. To the extent we've considered it, we assumed that everything was bad and so we should kill it all. And in fact, we know increasingly that many of these species are species we depend on for life, that we're in mutualisms with them, much as this midge is in a mutualism with the cacao plant. How many of you like chocolate? You depend on this mutualism. This fly pollinates the chocolate tree's flowers. In the same way, we have many mutualisms in our house. But we need to think about, are we favoring them or disfavoring them? And we've started to see a whole series of cases in which we have evidence. What was funny here? I'll have to think about that later. In which we have evidence that if we compare places that are more urban or less urban, or farms or not farms, that we're starting to see evidence of a whole series of chronic autoimmune disorders associated, it appears, with the loss of specific kinds of microbes, or whole groups of microbes, or even in some cases worms. That where those go missing, a bunch of problems emerge. And one of the classic examples of this is in the border of Finland and Russia, where the same group of people, genetically very similar, before World War II, lived in the same way together. After World War II, were separated by the boundary between Russia and Finland. After World War II, their trajectory diverged, and on the Finnish side, these communities became very developed, lots of cement, big houses, big closed doors, lots of good public health, and on the Russian side, not so much. And in that very short time, we see very different health outcomes in the two regions. And so in Finland, all of the autoimmune disorders, including allergies that are becoming problems in developed countries in general, became more common. In Russia, they didn't. And pardon that this figure is a little hard to read. And you can see this for birch pollen, but it's true for lots of the things that have been measured. And so the question becomes, what's going on? And so the argument in the literature is that what's happened here is that some of the key microbes that the people in Finland needed were cut off from the daily lives of the people living in Finland. We got rid of them. We cleaned too much out of the house. And all of these problems emerged as a result. And so we don't really understand this very well yet, but it looks like this is clearly part of what's going on there, and it's part of the same kind of story that's happening in Houston. How many of you in here know somebody with Crohn's disease? Inflammatory bowel, allergies, asthma. Yeah, so these are all problems we didn't deal with in the same numbers a couple of decades ago. And so the argument is that it's something about this transition, the cleanness of our indoor environments and our exposures that's associated with this change. And we can go into that for hours and hours, but I'm interested in a particular part of this, which is that if this is true, it actually makes a pretty specific hypothesis about the species we used to be exposed to. It suggests that there used to be more diversity, that diversity is good, that we've lost diversity, and that we've also lost specific beneficial species. And so implicitly, this means if we were to study traditional houses, we should see those things. But the interesting thing is that nobody studied traditional houses. And so we started to go about moving around the world to study traditional houses to see what lived in them. And so we worked with the Himba and we had families swab their houses just like we would in New Jersey, to look at whether these traditional houses, which are they're modern houses, but they look much more like the first houses we know about than they look like a house in New Jersey. And so what we imagine is that maybe the microbial environment of these houses is more like our ancestral environment too. And so what we would predict is that these houses would be more diverse, they'd have more environmental microbes, and there'd be less missing of whatever it is that we need. And so we sampled these houses with families. This is that same kind of map. Oh, that is me. And what we see here is, in fact, the Himba houses are very different, and that the reason that they're different is because they have a bunch of environmental microbes that are not in the houses in the US that we've sampled. What's cool is if you look in the Himba houses, there's still structure by environment, and so the pillowcase is still a little bit different because it's getting more skin microbes, even though the scale is small. We also see that those houses are much more diverse, more kinds of microbes in the houses for the same habitat types. And so that's cool. That's the first evidence of this sort of pattern. But what's interesting is if we're thinking about this being like our historical exposure to microbes, the reality is that most of the ways that our immune system works evolved not 20,000 years ago, evolved not in the context of these houses, but actually 1 million years ago or 10 million years ago. And so we also need to think about what were our exposures in other sorts of environments. And so we've started working in the homes of all kinds of mammals. We can't get prairie dogs to swab themselves. Yeah, it's the thumb thing. But I'll talk tonight about one of the things we've made the most advances in, and it's in the context of studying chimps and thinking about chimp nests and thinking about ape nests in general. And Jane Goodall, working with the Leakey Foundation, was the first to publish on chimp nests. And she was able to show that chimps make nightly nests, that they move from one nest to another nest the next night. And the interpretation was they were being abandoned so that species didn't build up in them. But what's neat is there's not been a lot of work on whether or not species build up in them. And so we can actually think about what lives in a chimp nest and how does it compare to what lives in a home. How does Jane Goodall's nest in this picture compare to the chimp's exposure? And it's worth saying that this is one axis of the many, many differences in our interactions with other species relative to the chimp's life and relative to primates' lives in general. And so we know, for example, that there are other sorts of things we've lost. Our kids rarely get eaten by eagles, at least where I'm from. I know in Texas things are bigger, but we also don't too often get eaten by leopards. We don't too often get eaten by snakes. And yet that's something that's changed. But few would argue that that mismatch is super important. The one context in which it's important is that our brains still evolved in the context of that history. And so even though no one's urging leopards on playgrounds, our bodies still make more sense in the light of that history. And so we're thinking about this, too, in the context of chimpanzee nests. And so this is Megan Temis, who works sometimes with mummies and then sometimes with chimpanzee nests. And so she teamed up with Fiona Stewart to work in the Isa Valley to sample chimp nests to figure out what was living in them. And this nest has Fiona living in it. She studied whether or not the nests were comfortable. The answer was sorta. But once she was no longer a graduate student, she never slept in one again. But we got Fiona and some of her assistants to sample chimpanzee nests, and we could then compare them to houses. And so on the one hand, our expectation was that the chimp nests would be much more diverse than this room is. We also thought they'd have more environmental microbes. But the chimps don't wear pants. And so it seemed maybe like there'd be a lot of fecal microbes too. We weren't sure what to expect. And we thought sure there'd be tons of arthropods associated with the chimps. And so we looked, and what we found was no arthropods associated with the chimps in their nests. None of their parasites were there. And a crazy signature of the outside environment. And so mostly this nest, in terms of microbes, is leaf microbes It's microbes associated with tree branches. It's the outdoors, and it varies season to season. We actually see almost no signature of chimpanzee in these nests. And so if we want to think about this, the proportion of microbes from bodies in a house in the US is about 30%. A Himba house is lower, and then a chimp nest is very small. And so in addition to having lost microbes during this transition, we also made a change where we now live with fecal microbes more so than at any other moment in history. And for daycares, our data suggests that 50% of the microbes in a daycare are fecal. And so that's a big change. And so if we want to think about what have we done to change our microbial environment, well, mostly we've made a lot more of ourselves. Like all of our houses look like we exploded in them. And the more you're living in an apartment, the more true that is. And then we got rid of all the stuff that's potentially beneficial. And so that's a pretty dopey thing to do, and yet that's the moment at which we are in our story. And so every day you breathe in thousands of species. You just did. Everybody take a gulp. Thousands of species. In addition, you will fail to be exposed to most of the species your ancestors breathed in and slept on. Some of each group can kill you. The cough was poorly timed. Most are benign. Some you depend on for life, and we can't really distinguish these groups yet. And so for the 10th graders thinking about what you might do, adults are so dumb that we can't tell whether the stuff you're breathing in is really bad for you or absolutely necessary. And so we're going to depend on you to save us from ourselves in many, many ways. So please do great things. You have the possibility to do them like never before. And so in the context of this, we're really at this really inauspicious moment where we know we live with 200,000 species, and we mostly don't know what they are or what they do. We do know some things. We know about some of the bad species. We know to get rid of bad species, vaccinate, wash your hands, use a toilet. We know to avoid favoring bad species, to don't overuse antibiotics, don't use antimicrobials, don't use antimicrobials. My university just made underpants with antimicrobials in them. I don't even know what that does, but it's just a bad deal. But the take home in this context is that we imagine our houses to be sterile. The space station is supposed to be sterile. It's so not sterile that you could actually probably see the microbes floating in that video if you look carefully. Our houses, the space station, are full of hundreds of thousands of species. Most are not well studied. Overuse of antibiotics, use of antimicrobials favors bad species, and favoring beneficial species will take much more science. And so what I hope to be able to say always in a talk like this is that we should redesign the home, that we should redesign our daily lives to favor the things that we need. We need to be able to build gardens of beneficial species. But I think we're not there yet, that we don't quite know what to do, which is ironic because the one thing that does know what to do in some contexts is our unconscious body. And so how many of you had your appendix out? All right, so students in the front, what's the appendix do? Nothing. Digestion? Nothing. Getting worse? So it turns out it's totally not, that's wrong. And Bill Parker at Duke Medical Center a little while ago was in his lab spinning around thinking about the appendix. Randall Bollinger, who's a surgeon, was there with him. And Bill says, "Randall, you know what, I figured out the appendix." And what Bill thought was that maybe the appendix is a storage organ for good bacteria. And if you lose your good bacteria because you get a bad gut infection, that they can be recolonized from your appendix. And I knew Bill at this time, and he said, "I'm writing a paper about it, it's going to be great." And I thought, Bill, people are going to call you an idiot. But he's a character, he didn't care. And so he published this paper, and I thought immediately there'd be a response from all the people that study the appendix. And there wasn't, because basically nobody studies the appendix. Because it does nothing. But Bill said, well, you know, the real test would be if you went someplace where people still get really severe gut infections, the people who have their appendix should be more likely to recover from those infections. But this was a problem for a variety of reasons. Nobody was ever going to do that study. And then I was writing about Bill's work, and I found this paper from people at the University of Rochester. And they were studying Clostridium difficile, which is a really bad bacteria that, especially in hospital contexts, if you've taken a lot of antibiotics, can take over the gut, and it's like a weed. It grows back faster than your good microbes grow back. And so it can be deadly, and probably people on their own have lost family members because of C. diff. Well, they did a study of recovery of C. diff, from C. diff, and they just happened to note as one of their data things, do people have an appendix or not? It was already on the sheet. And people who had an appendix were 4 times more likely to recover from C. diff, which is to suggest that Bill might just be right, which is crazy because it's an organ. We're not still supposed to be explaining what organs do. And yet that's where we are in this moment. And so to me, this is fascinating because it suggests that our bodies evolved the ability to favor beneficial microbes, even though our conscious brains don't even know which ones are beneficial. Another related phenomenon, stomach pH. In textbooks, we mostly learn that your stomach is acidic to break down protein. But it turns out that if you look across mammals, and I've lost my labels here at the bottom, so I'll have to walk you through it, that stomach pH varies widely among mammals and birds, and that it appears not to have to do solely with diet, but with your risk of pathogens. And so it looks like what may be going on with stomach pH is that you have a really acidic stomach if the risk of eating pathogens is high. And the stomach is then a filter for killing off pathogens. And so there's a fair amount of evidence for this. But it's another way in which our body might be filtering one kind of microbe relative to another, which to me is fascinating. Also fascinating is that medically we often change the pH of the stomach with gastric bypass, proton pumps. And when we do that, we now see that the gut microbes change, often in negative ways, which is what we would expect if the stomach was controlling what colonizes and what doesn't. Another example, Aspen Reese at Duke University has recently shown that the body can selectively feed microbes in the gut in response to what it needs. And so if there are a lot of really hard to break down carbohydrates in the gut, the body actually produces more mucin, which feeds the microbes. And this works because the microbes are really nitrogen limited. They need nitrogen way more than we do. But by the time your food gets to them, the nitrogen's mostly gone. And so the body uses doses of nitrogen to control who it wants to do what. And so our bodies are sophisticated in all these things. Another example, we've been studying armpits. I should just leave there. But it turns out they're fascinating. And they're fascinating because they have these really enlarged organs called axillary organs that are bundles of apocrine glands, which we describe as sweat glands, but they don't produce sweat. They produce food for microbes. And they do it in response to stressful situations. The genes that control those glands are under stronger natural selection than any genes anybody's documented in humans. And we don't understand why. What we have been able to figure out is that these glands produce really costly food, and the microbes that they favor are different in different primates. And so there you see the baboons, the rhesus macaques on the right. They're super diverse, and they're pretty covered in fecal microbes. Have you hugged a baboon today is never going to be a good ad. The gorillas and the chimps, each one of these bars is a different kind of microbe. They're really dominated by Corynebacterium. That's what you have in your armpit that's stinky. Really slow growing, hard to grow in the lab. That's their dominant armpit bacteria and on their fur in general. Humans, that's the case for some of us, but not all of us, and we couldn't explain this forever. And the only reason we started this study is because I met a zookeeper who said that she used to work with gorillas, that she now smells like a gorilla, it makes her dating life terrible. Can we explain it? And we can't explain it, but this was a fun study anyway. It turns out that humans are really, really variable, and what's going on is antiperspirant use. When you use antiperspirant, you disfavor Corynebacterium, and you favor Staphylococcus. That's crazy because that's what we think of as the medically normal skin microbe. And its normalness is partially a function of the products we use. The other interesting thing is there's a single gene, one nucleotide, one little teeny bit of one gene, and that gene explains another 40% of the variation in person-to-person in skin microbes. Single nucleotide. We can talk more about it later, but it's pretty amazing. And so we're working more on this. But the point being, our bodies do a lot to favor specific microbes. This is relative to what we do medically. This is like the laughs in this audience relative to when I think they're coming are wonderfully unpredictable. Well, I guess that's funny. So fecal transplants, when I wrote The Wildlife of Our Bodies not very long ago, were a radical and ridiculous thing that nobody was ever going to do. That's not a very old book. Now, if you are a medical resident at the University of North Carolina, you are going to be a feces donor. Almost certainly. Because fecal transplants have become common for a variety of problems, but especially for seed death. They're amazing. They often work. And this is really fascinating medically because we're restoring ecosystems. But the humble element of this is that what we're doing is we're going to somebody who looks healthy, and we say, "You look healthy. I bet you have healthy shit." We're going to give it to this person who doesn't. And so that's how it works. And so what we would hope for in this moment is we would be able to figure out what one person is missing. We'd be able to figure out the perfect cocktail of microbes. But we can't. Nobody's made that work. And so instead, we just take like the whole rainforest of stuff from a healthy person, and we transfer it. That's amazing. But that's the moment in which we find ourselves. Similarly, there have not been a ton of studies of the total microbial composition of hospitals because everybody's afraid they'll get sued. But in one of the nicest experiments of the microbes in hospitals, a group at Oregon compared what happened in hospital rooms when you left the windows open to when you didn't. And they were able to show that the total load of bacteria didn't change. It was about the same. But the proportion of those bacteria that were pathogens declined. Again, that's crazy. That we're so early in the moment of understanding all of this that opening a window in a hospital is a healthier thing than where we are right now. And there are lots of details to work out, but very few people are studying them. Oh, I'm one behind. I'm sorry. Maybe that's why it was funny. And so what I want to be able to do in a talk like this in a few years is now to, at this point, list the species that you should go home and eat. List the species you should go home and favor. List the probiotics that are the best to take. And we're getting there, but we're not quite there yet. But one of the interesting possibilities is that there are species that benefit us not just in the short term, but also in the long term. And so Michelle, who you see here, whose parents are in the audience, where are you guys? There you are. No, somebody else wave their. You just got parentally scooped. But so Michelle started this amazing project to sample all of the arthropods in homes. And so what Michelle found was that there are, just like with the bacteria, there are way more kinds of arthropods living in your houses than you might imagine. 2,000 species and counting. This is more intensive. It takes having this guy and other people go down into basements and check things out. But there are lots of flies and beetles and ants, lots of species of beetles, a bunch of crazy stuff, like this is a spitting spider that lives indoors and not outdoors. It spits a ball of silk on its prey. There's one in my house, just walks around spitting a ball of silk on things. This is the world's smallest cricket. It's also in my house. I don't know why. We find a wasp that lays its eggs in the bodies of roaches, a teeny tiny wasp. It's not named, super common. And we can do all kinds of cool ecology with these data that Michelle's been collecting. So you can look at like how the number of doors you have affects the number of kinds of arthropods you have. But the other thing we can do is go one by one and study the biology of these species. And we do this with the public. And so this is a camel cricket. And at one point we ask people, do you have camel crickets in your house? Anybody here have them? They look like that. So they're not quite this big. So we asked people, and this is the map we got in one day. So red dots, people who said they had them. White dots, people who said they didn't. And what we knew as scientists was that this was totally wrong. And so maybe people in California were like scared to go in their basements. I don't know. And so we then asked for pictures. And we were in for a total surprise because instead of that native camel cricket we expected, most of these records were of a giant invasive Japanese camel cricket that had moved basement to basement totally unnoticed. No scientist knew it was there. We now estimate numbers in the tens of millions. And then in looking for it, we found a second species of introduced camel cricket nobody knew was there. And so this was super amazing, and I went and talked to people again and again about this, and to me it's like, oh, cool biology. And again, people had the same response. Can you guess it? Yeah, something like that. Or what good is it? Or how do I squish it? And so this, as a biologist, this is a sad thing because I love these species. But I thought, okay, what good might it be? And so I started thinking, and these are really cave animals. And cave animals live in really weird environments. They live in the dark, they lose a lot of their pigment, and they typically eat really low nutrient foods. And they're not just low nitrogen or phosphorus, there's actually just not much carbon even. And so what if cave crickets, like camel crickets, like other cave animals, have microbes in their guts that can break down things that are almost impossible to break down because they have to, because they live in caves. And so I thought, well, I don't know how to study that, but I know Amy Grundon, who's in the next building, and whose dad was like the chief lie detector during the Cold War in Germany. It's an interesting story. But I mean, she's super hard to read if she's excited about her project. And she said, well, if you could find microbes that would break down this from the camel crickets, that would be cool. This is black liquor. It's the end product of making paper. It's the waste. And it's mostly lignin in an acid bath. And I was like, oh, that sounds awesome. Let's do it. What Amy didn't tell me is that there were only 4 bacteria species on Earth that break down lignin, and none do it very well. And so it was like the hardest thing ever to imagine we were going to get a win out of. And yet, we found an additional 8 species that break down lignin, and all of them can do it in an acid bath. And so we're now trying to get these microbes to make energy out of the waste from paper pulp. And so this is a benefit of these species in your house. It's not in the short term, but it's the value of biodiversity in the longer term. Another example was in Tenerife with my family, and my daughter was down at the shore, and she found this salt accumulating on the rocks. And so she started asking, because she's a biologist's daughter, I wonder what's growing in this salt. And I realized, I didn't know either. Nobody knew, it turned out. And so the cool thing about that is that you have a lab, you can go home and start studying it. And so we started studying the biology of salt and salt shakers from around the world. Super interesting, different in different places, totally useless but fun. And then I went across the hallway. And this guy, Ben Reading, we just hired him, and he works on sturgeon and caviar. And I'm interested in almost everything except fish. But we're talking to Ben, and Ben was talking about with caviar, there's a huge problem that's really variable from one batch to the next. And caviar is in a salt brine. And so we started talking about, well, what if you could use the microbes we find in the salt shakers to improve the caviar? And we've just found a yeast that we think can stabilize the flavor of the caviar and keep it from spoiling from salt shakers in houses. Another example, I was in my basement, same one where that teeny cricket was, doing laundry, and I get a phone call like 11:30 at night. It says, Rob, yeah, if we give you a box and it had dust in it. Could you tell us where the box was from? I thought, I'm doing my laundry. This is a weird question. I said, yeah, I bet we could figure that out based on the DNA of what was in it. And I said, well, who is this? And they said, it's a government agency. I'm like, oh. And so it turned out it was a government agency, and so we actually tried to do this. And it turns out that we can do it. That if we know where a sample comes from of dust, well, if we know the fungi on a sample of dust, we can predict where that sample came from within 100 kilometers. On the West Coast, within 50 kilometers. And so we're now using this to figure out if we can track where IEDs have come from, based on the dust that's under the tape on the IEDs. And that's all because I was in my basement looking for crickets and doing laundry. Another example, work with Michelle on the space mite project. These are these mites that live in the faces of all adults. You guys might not have them yet, which is another reason to stay clear of adults. Most adults have 2 species. They have sex on your head, and then they fill up with feces and food, and they don't have an anus, and so they just disintegrate. So they're kind of a weird thing, but they're incredibly abundant. Nobody studies them very much. And we think that there's, 17, trillion? How many zeros do I have? Of these mites on people's heads. And so we started thinking, what use could they be? Well, what's interesting is most of the time you generate no immune response to them, which made us start to think that these are almost certainly producing immunosuppressants. And so we're now looking at these super common animals for new immunosuppressants to be used. Another example, all those microbes on your skin, on your armpits, in your belly button, they're what other people smell when they come near you. It's what you smell of that person you don't know that's too close to you right now. They're what mosquitoes smell when they get close to you. They're also, and that's what mosquitoes use to choose whether to bite you, and they're also what viruses and bacterial pathogens first encounter when they land on you. And so which microbes you have on your skin is hugely important. And so we're now working to figure out, can we actually develop microbial probiotics that will be a deterrent to mosquitoes? Again, in the context of stuff we do in backyards. Another example, and this is the last one I'm going to wrap up, is that we've been studying the exoskeletons of ants. And they're really cool. That's just their outside, right? And we figured out that it looks like there's an interaction between what their skeleton looks like and the compounds they produce to kill microbes. And so we thought, could we use what we learned from that to produce new kinds of textiles? And so we talked to people in textiles, talked to people in design, and we didn't quite get all the way to new textiles, but we made new pillows based on the designs from ant exoskeletons. And so you don't always get what you think you're going to get. But in thinking about the species all around us, and this is a species from a house, we see new things. And finally, the last example, and we have a bunch more, we found a microbe in tap water and gutters that precipitates gold. And we're now working on that. That's just crazy, right? But it's true. And so to conclude, we confront at this moment, I think, the astonishing potential value of the species we haven't yet studied. Species we can sometimes find and study in homes around the world, but we need to conserve wherever they are because we don't know which ones of these are beneficial yet. And this is a crazy reality, that until we figure out which of these species in our homes we need, in our backyards we need, in the forests around us we need to keep them. And so while it may seem weird to have to save microbes, we do. We have to save all of this stuff. And so this is the point of the talk as I close that you probably want to know, can't you name at least one household microbe we know is good in the short term? And we have one, so we found a new yeast from around houses that makes a really good beer, and that's good in the short term. But the other, and this goes back to schools, is sourdough. Because it turns out when you make sourdough, it relies on the microbes that are living in your home. So you put out a mix of some grain ground up and water, sometimes some other ingredients, and you produce a microbial mixture that eventually produces lactic acid and can make bread rise. And this is a magical thing we only partially understand. And this happens as a function of the microbes in the wheat, on your skin, and in your house. And so which things live in your house almost certainly affect what kind of sourdough starter you can make. And so we've just launched a new study, and so if you have sourdough starters, we're studying starters from around the world. We're listening to people tell their stories about their starters, and we're trying to understand how much do the microbes in your house matter to your starter? How much do the microbes in your skin matter? How much does the climate matter? How much does the grain that you use matter? And we're going to grow all those microbes up, and then in July we go to Belgium, to the Paratos facility where they make a bunch of breads, and we're going to use those different starters to make breads using the same ingredients, and then we're going to bake 1,000 breads and have a taste-off to try to understand the flavors of individual homes and bodies and what those contribute to us. And so maybe it doesn't save us from disaster, but it saves us from hunger. And I'm happy to close there. So thank you very much for having me.